Good morning, everyone. And thank you very much for joining this session. See, there are still some, let's say, quite a bunch of people here interested by this very technical topic. So we might wonder why so many people are interested by allergen. Obviously, allergen remain a major concern for food industry. Firstly, because it raises significant and, let's say, serious health issue to consumers, and consequently, legal, reputational, financial risk to businesses. Moreover, I would say that it's fair to say that allergen is among the most complex topic when it comes to food safety. And in the past years, science and regulation have continuously, significantly evolved in this field. So setting up a very, let's say, effective allergen management program is critical for food business operators to address those challenges. And we have to have a very robust and structured approach, integrating prevention, control, verification, communication, for food business operators to protect the consumers. This is really critical. So in this session, we will explore all the components of a comprehensive allergen management program for food business operators with some very, let's say, specific focus on risk assessment methodologies and precautionary allergen management. Labeling. So in order to deep into those topics, I'm really privileged to be joined by three scientists internationally recognized for their deep knowledge and expertise in the field. So we have with us René Crevel, Neil Buck, and Jasmine Lacey-Slee. I will start by introducing René. We will make our first talk, and you will see how impressive the bio of René is to make it super legitimate to talk about risk assessment methodology. So René runs currently a consultancy centered on food allergen today. Before he made his career in Unilever, being the reference for risk assessment and risk management, served as a UK Committee on Toxicity, has been also scientific advisor to ILSI Europe, Food Allergy Task Force, and finally was a member of the FAO-WHO expert consultation on risk assessment of food allergens. So now, René, please, the floor is yours. You're going to bring us through the scientific foundation of allergen risk assessment, commenting on reference doses and their application to precautionary allergen labeling.
Thank you very much for your introduction, actually. It's very nice. Saves me having to say a lot of things, actually, about that. So what I'm going to tell you about is, or at least some aspects, at least, of the scientific foundation of risk assessment, specifically the development of reference doses and the application to PAL in the context of the FAO-WHO consultation, which took place from about the end of 2020 to 2023, more or less. I think we finished the reports by about then. So, which were set by Codex, which I won't go through in detail. You will be able to have the slides later anyway. Was in five parts, actually. Originally, it was going to be 3, as it happens. It was going to run over 3 weeks for each of them. And I'm going to focus, of course, on part 2, review and establish threshold levels in foods for the priority allergens. And I make no apologies for the grammar. That was the FAO-WHO grammar, anyway. And part 3, of course, establishing precautionary allergen labeling, actually. I was privileged, actually, as Monique mentioned, to be a member of the FAO-WHO consultation, and in fact, more privileged to chair part 2, the meetings associated with part 2, and finally the report and so on. Okay, so let's go through that part. I'll go through that. Now, the terms of reference which Codex gave us, well, we thought actually it was a very good idea, to try and stick as closely as possible to them and see what they actually want out of the consultation. And it's quite clear here that they're actually asking for risk-based things. They're talking about thresholds below which the majority of allergic consumers would not suffer an adverse reaction, not below which no one will suffer anything at all, will experience anything at all. That's quite a different demand. They also asked about an acceptable level of allergic reaction risk, which does not negatively impact public health. And we took those characteristics into consideration when developing the reference doses. They also pointed us very kindly as well, again, to, in terms of reference to this particular IPCS monograph, and from reading the relevant chapter, it's pretty clear that we're looking for health-based guidance values, as defined in that monograph, which represent exposure without appreciable health risk. And so we examined the different approaches and decided that those which were based on dose distribution modeling actually were the ones. So dose distribution modeling, for anyone, well, probably not many who don't know something about it actually, describes the proportion of people who react to a particular amount, a dose of allergen in a controlled clinical challenge. So, for instance, in ED05, as a population, for the population, is the value below which 95% would not react, actually, or 5% would react, actually, to take the opposite side of it. Right. This just describes the flow chart for all of through which we proceeded to derive that, and I don't think I can. Yes, I can read some of it. Yes. So whoops, if I can manage to do that. Whoops. No. Yes. Particularly, I'm going to say something about the choice of approach, then the definition of the safety objective, because those are important in my context. So we spent quite a long time. I've already talked about the previous one, I just realized. We spent quite a long time, actually, developing this safety objective, and I've pointed to the critical words, highlighted them in red in this particular sentence, actually. So to minimize to a point where further refinement does not meaningfully reduce the health impact, the probability of any clinically relevant objective allergic response. That took several meetings, meetings that way, as defined by dose distribution modeling, so that's what we are using. And also, of course, supported by data regarding severity of symptoms in the likely range of envisioned reference doses. And of course, we also have to take into other considerations, data quantity, quality, and availability, and so on, which I'll talk about in the next slide. Yes. So in terms of data considerations, the data that you use for defining dose distributions, the quality considerations for those are representativeness of the population tested, the bias. Now, that's quite difficult, actually, to obtain a totally representative population. But I think, actually, these are based on the double-blind, placebo-controlled food challenges in people who have been diagnosed with a food allergy. So they are probably the bias, if anything, is probably a positive towards a higher value than than a lower one. The number of data points is very important. I've mentioned number of left and right censored values. For those who are not familiar with those particular terms from statistics, left censoring means someone reacts at the first dose. So you don't really know what their threshold is, below which they don't react at all. Right censored, they don't react at the top dose within the challenge. Both of those will affect the dose distribution and make it skew it, actually. And that is a problem that people need to be aware of. So, for instance, actually, and we have looked within the database that I'm talking about at, for instance, results from people who are being screened for participation in immunotherapy trial, and find actually, yes, most of they are mostly right censored. So they don't actually, they don't want people Yeah, they don't want people who actually react at higher doses. And that skews ED05, that can be easily demonstrated. Now, availability, access considerations, data quantity, of course, that's very important. Do they cover all the allergenic foods? That's for the purpose of the consultation. And the data quality, what are the inclusion and exclusion criteria? How are minimum eliciting doses actually extracted from the data for modeling? And then availability and access. And we concluded, after some debate, but not too much debate because there aren't many alternatives, that the data set reported in the publications indicated there, which I think are free access, if I remember correctly, were best meet the criteria. Certainly, they're the most comprehensive and best described. Now, I think currently they have about 3,500 data points, actually, for covering quite a wide range of allergens, including all the priority ones. Yes, the quality criteria actually described in considerable detail in the publication, which is listed here. The only downside is, for reasons which I have no time to go into, and I'm not necessarily completely familiar with, because it's not my database or one in which I participate that way, they're not publicly available. That is potentially an issue. Right. Now, as I mentioned, we needed to characterize, in considering what reference doses we come to, we needed to characterize the reactions at proposed reference doses. Now, it was very easy to say, well, okay, we're only going to look at the lower end, of course, of doses that provoke reactions. We eliminated ED10. We thought actually identifying ED10, well, using ED10 would actually not provide the right degree of protection. So we were left with ED01 and ED05. For practical purposes, it would not be possible to go below ED01 because you just don't have enough data points within that as you go to, say, ED 0.01 or anything of that sort. We had a choice between those two. And the interesting thing, actually, which you probably can't see on there because is one characteristic, oops, sorry, I'll go back again, press the wrong thing, is that in this particular study, actually, meta-analysis, I should say, the authors actually were looking at the proportion of individuals who reacted when they were challenged at approximately ED05 and ED01, who experienced mild anaphylaxis, and I'll tell you a little bit more about that. And in fact, interestingly enough, they found that whether it was ED01 or ED05, it was actually the same proportion. Of course, because more people actually reacting at ED05, then there are more people as a whole number. So that's one of the characteristic aspects. Right. Now, anaphylaxis, yes, can be an emotive term, and the World Allergy Organization actually was very careful to, when it reviewed that particular phenomenon, to define it very carefully. And as you can see here, one part of the definition is typical skin symptoms and significant symptoms from at least one other organ system, or for instance, cardiovascular, respiratory, or something else, or exposure to a probable allergen for that patient with respiratory or cardiovascular compromise. And what I'd like to point you to is the statement here, below, there, that the literature indicates that fatal and near-fatal events are rare, and I'll show you some data for that, even when not appropriately treated, which are actually a surprise to many of us who are not clinicians. Therefore, the majority of anaphylaxis reactions cannot be described as life-threatening in themselves. Something to bear in mind when we're talking about the criteria here that we used. Whoops, did it go? No, there we are. And you can see here, actually, even better illustrated in this particular thing, you've got the people who react at ED05 and below, obviously. You can see anaphylaxis here, at least 80% resolve without treatment. Oops, without treatment. And the remainder usually respond to a single dose of adrenaline. Severe anaphylaxis is confined to that, and fatal even lower. And those are the estimates that we took into account in deciding whether the reference ED05 is satisfactory. And very recently, actually, this is a 2025, published in 2020, in January 2025, actually, the custodians of the FARP-TNO allergen threshold database, as it's now called, actually, reviewed the severity of symptoms associated with food challenge with doses up to and including ED10. And what they found there, as you can see, is of 1,092 symptoms recorded at 444 food challenges in the 101 patients, and only 2, and both of a respiratory nature actually, were characterized as severe, and those occurred not at ED05, and certainly not below ED05, but at ED08 approximately. And symptoms at ED05 were of a mild or moderate severity, which actually is the title of the paper, in fact. So that's providing further evidence of the appropriateness. Now, based on all these, yes, we didn't just, in fact, I mean, we were even more precautionary in some ways in how we propose the reference doses, because the reference doses are derived from ED05, but we simplified things, so we have actually fewer numbers, and which hopefully will make it, a bit more, a bit easier for risk managers when assessing the risk from for PAL purposes. Right, that's one. There were further recommendations, actually. One was to standardize analytical results by expressing them as milligram of protein from the allergenic source per kilogram of the product analyzed. Now, that might seem quite, compared to some of the other science, quite a straightforward thing, actually, but there are many ways in which results are reported, and of course that makes it more difficult for those who need to use those results to apply them. And also actually because we recognized that analytical methods were not necessarily all up to the task to apply, to propose, I should say, a default uncertainty factor of 3 times to the claimed limit of detection. So that, yes, okay. So now I'll say something about the precautionary allergen labeling. There's only a couple of slides, so it should be, yeah, should be okay. Right. We drew a number of conclusions about, before we embarking on how we should use reference doses in the context of PAL. One, that precautionary allergen labeling can be an effective tool, strategy to provide, to protect consumers from unintended allergen presence. And reference doses, derived as we indicated, would be helpful in ensuring that protection and providing a better system for consumers. But we noted also that the current use of PAL is voluntary and is often not part of a standardized risk assessment process. And consumers get very confused about it. They get confused about it, and it is not surprising, actually. I think you'll probably all know that. And the available evidence actually indicates that some manufacturers, consumers, and other stakeholders do not understand current strategies to communicate precautionary messages relating to risks posed by UAPN products. So those are the things. So how do we, what did we propose? Well, we mandated, we suggested that the use of risk assessments for application of PAL should be mandated. Now, it sounds very regulatory, actually. It could be interpreted differently. I mean, there may be, it may not be based on regulation. That's a matter for debate, and I've heard some of the debate here today during the conference. It should be based on reference doses, obviously established by the consultation, in the consultation, makes sense, and provide consumers with an indication that the risk assessment has been performed. Now, the important thing, actually, one critical thing, actually, is, we pointed out, I mean, I'm sure it will be obvious to all of you, because you are, well, educated, this is appropriate quality, well, appropriate prerequisite programs of quality control, hygiene, and risk mitigation practices to minimize UAP should be in place. No point in starting your risk assessment until you've actually got those in place. And then, of course, it's very straightforward. You've got, either you have a conclusion that the risk is negligible because the amount of the concentration of unintended allergen is below the action level, or vice versa, it isn't, and then it isn't. Then the risk can't be excluded, and you use PAL. Now there are lots of other things to consider in terms of how you apply those aspects, and my colleague Neil will be talking to you, I think, about that. Right. Okay. Oh yes, that's it. Thank you for your attention.
Thank you very much. Thank you very much, René. So, back to my introduction. So, that was a very nice intervention to illustrate how much science has been evolving for the past years. Now, I would like to move to the regulatory evolution, handing over to Neil, who will discuss the evolving regulatory landscape, highlighting maybe how industry might be behind now the regulatory expectations. So, Neil, just a few words about you, and if you want to comment, please feel free. So, Neil is a corporate toxicologist with General Mills for now close to 25 years, active in various stakeholder fora in Europe and US. And please comment further if you wish. Thank you very much, Neil.
Nope, c'est bon. Thank you very much.
C'est bon.
Yes, so I'm going to talk about where we are at this point in time in terms of the road to harmonization across the wider food industry. So the application of this new tool across the wider industry, and some of the challenges that we face at this point in time and how we might overcome those challenges. So in particular, as Monique mentioned, there is an emerging disconnect at this point in time in terms of authoritative expectations around the need for industry to apply this new tool, and actually the reality of the preparedness of the wider industry. So what I'll talk about is also one of the main challenges, one of the main technical challenges underlying the need for effective application and a hurdle to effective application, and talk about next steps. So of course, quantitative risk assessment has a number of possible roles in terms of the wider aspects of allergen management, and could be used in terms of understanding the impact on populations in terms of helping to assess incidents when incidents happen on market, or indeed decisions on exemptions from precautionary labelling. But the biggest impact day to day will be on decisions for precautionary labelling, whether precautionary labelling is appropriate or not. So actually, that represents a step change in complexity, because the situation that we have today and historically in terms of managing precautionary labelling is based on a qualitative approach. So essentially a binary approach of whether an allergen may be present or is not present. And that information is communicated across the supply chain and ultimately to consumers. So when we're talking about quantitative risk assessment on top of the qualitative approach, there's more information that needs to be communicated across supply chains, and that information then needs to be used in an assessment by the finished food operator to help determine whether precautionary labelling is appropriate or not. And so that is a step change in complexity in terms of comparison to what the current situation is today and historically. So fortunately, we have some very good technical guidance documents that are now available, that have been developed by a wide stakeholder community and have been developed, managed by the ILSI organization. And you may have found on your chairs, there are some flyers there, and also there's a QR code that links to the website where this technical guidance is available. So that's about understanding what data is needed and the mechanics of doing the assessment. So in terms of understanding where that assessment fits in the wider decision-making process of whether precautionary labelling is appropriate or not, there has also been some progress made. So we are in the process at the moment of aligning across industry on the basic principles of how QRA should fit into that decision-making process. So very simply, there are three fundamental steps. It's really about understanding the points of cross-contact that are in your supply chains, whether those points of cross-contact can be fully mitigated, and if not, if there remains a chance of occurrence of cross-contact within a supplied material, within a foodstuff, whether or not it's possible to positively demonstrate a reasonable certainty of no harm to consumers. If it's possible to demonstrate reasonable certainty of no harm to consumers, then precautionary labeling is not required. And that's where QRA fits in. And there's a number of other things that fit into that process in step 2. And if it's not possible to positively demonstrate reasonable certainty of no harm to allergic consumers, then the third step is to consider precautionary labeling. And there's schemes that have been developed to try and communicate around this approach. So where are we at this point in time is that it's recognized that there needs to be a wider industry awareness and application of the technique to really benefit allergic consumers. There's no benefit for allergic consumers unless all of the products in the supermarket are using the same methodology to determine whether precautionary labeling is appropriate or not. But in reality, where we are at this point is that it's only corners of our industry that actually have experience and are systematically using the methodology. So there's very little take-up amongst the wider industry, or even awareness or training amongst the wider industry. Now, that is actually juxtaposed at this point in time by emerging regulations, particularly in Europe and through the Codex Alimentarius process, whereby there are authoritative expectations that industry will apply the approach. So there is an emerging disconnect between the preparedness of industry and the authoritative expectations. So just to summarize, there is a really strong rationale that the decision-making on precautionary labeling needs to be improved. That is pretty much globally recognized. And really the only way of doing that is by implementing quantitative risk assessment. Thankfully, due to the efforts of René, Jasmine, and other experts, there is a very strong scientific underpinning now in terms of allergen reference doses or allergen thresholds. We've made good progress in terms of agreeing when and how to perform allergen quantitative risk assessment. And there is now some progress in aligning on the place of QRA within the wider context of decision-making around allergen management. But there is this limited activation across the wider industry. And as mentioned, that is in contrast to these rapidly emerging authoritative expectations. Now, I would be so bold as to suggest that part of the reason why there is that rapidly emerging authority expectation is because the authorities themselves have got limited experience in the methodology, in particular around the fact that there are only specific situations where it is appropriate to apply quantitative risk assessment and to use that for decision-making purposes. And a lot of that appropriateness really revolves around the quality of data that is available to you from your supply chains. And this is a critical issue because when you're doing quantitative risk assessment, really what that is a comparison between the reference dose that René's just explained and the exposure to which the consumer will be subjected. So it's just that comparison. But actually, it can be quite complicated to get an accurate exposure assessment. And the problem with this is, if there is this emerging push from authoritative bodies, but there's limited experience across the wider industry, what that will result in is spurious and possibly incorrect decisions being made underlying precautionary labeling, incorrect calculations that are being done. So understanding the quality of the data that you have available is really critical to doing quantitative risk assessment. There's some slides in here for reference because they're available as part of the conference pack, but this really describes the situation on the decision making when there is sufficient and insufficient data available to you. So what do we really mean by sufficient quality of data? Sufficient quality of data enables you to make an exposure assessment which is reasonably worst case. So what does that mean? It's an exposure assessment that reflects reality, but does not underestimate the exposure to the allergen for the consumer. Why is that important? Well, in any exposure assessment, unless you're extremely lucky, you never actually identify what is the reality of the consumer exposure. It's always an estimate that is either below or above the reality of what the consumer is exposed to. And for quantitative risk assessment, the sweet spot, what you need to understand is, does the data support your desired estimate, which is a moderate overestimation of the exposure. So it's close to reality, but it doesn't underestimate. Because if you underestimate, that's potentially dangerous, because you may make a decision that precautionary labeling is not appropriate, when actually it may well be. So the data that feeds into your quantitative risk assessment is the form, the distribution, the concentration, and in some circumstances the frequency of how often the cross-contact occurs. And all of that data can carry overestimation and underestimation. And there's consequences to that in terms of what it means in terms of your exposure assessment. So understanding the quality of the available data is really critical. But it's not just you, as, for example, in my case, working for a finished food company. Your entire supply chain needs to understand this and needs to operate to the same standards and have the same level of experience. And so guidance is really required as a common source of guidance, and critical is training and experience. Fortunately, we do have a pretty good technical understanding of how to understand the quality of data that is available to you, how to kind of identify whether the quality of data is sufficient or not to use in risk assessment. However, that's in fairly technical documents at the moment, and it needs to be translated into something that's suitable for training to the wider industry. And just to summarize, is that both the qualitative and the quantitative approach have pros and cons, opportunities and challenges. So with the existing qualitative approach, of course, it's relatively simple to operate, and fortunately, it is deeply embedded across our industry. Whereas with the quantitative approach, it is a step change in complexity compared to the qualitative approach. And it may well be that there is some resistance to change across the wider industry on what is, of course, a critical safety topic. However, there is now strengthening pressure coming from authoritative bodies, in particular in Europe, to apply QRA. However, there has been limited awareness so far across the wider industry, and experience is really key. Understanding the quality of data that you have available to you, how to improve the quality of data, what kind of assessment is appropriate based on that quality of data that you have available to you. So where we are right now is that great progress has been made on technical guidances, particularly wide stakeholder alignment on the mechanics of how you use this tool. However, activation is needed across the wider industry, and that really needs standardization, guidance that's applied across the wider industry, and training, and most importantly, experience. And importantly, there needs to be a paradigm shift in how data is communicated between food operators, because we're talking about quite a different communication between the qualitative approach, where there is an allergen that may or may not be present, and the quantitative approach, which relies on more detailed information around concentration, form, distribution, etc. And so there needs to be a change, a significant change in how that information is captured and communicated. Okay. Oh, Jasmine.
Thank you so much, Neil. So I believe you made clear, and you put a little bit of pressure on food business operators, making clear that they must really leverage the scientific methodologies, risk approach at best, upgrading the industry standard in order to realign with the regulatory expectations.
Yes.
So that was very clear.
Indeed. Thank
So
you.
now we will hand over to Jasmine, who will bridge theory with practice by outlining practical consideration for implementing quantitative risk assessment. So Jasmine is very well known for her expertise in the allergen field. Just a few words about your background, Jasmine. So you are a food specialist, over 25 years of experience, having worked for different companies, Lactalis, Coca-Cola, Euro-Pacific Partners, and BVQ. Since 2021, Jasmine has been, let's say, the president of the Allergen Bureau that you will present in a second, and board chair. You are also part of the Australian Institute of Food Science and Technology Scientific and Advisory Committee. And last, you have served as representative of the AOAC Food Allergen Working Group. Quite a big, let's say, a very nice bio. So please, Jasmine, the floor is yours.
Thank you so much, Monique. It's an honour to be here. I am going to, I guess, dive in a different direction. So we've listened to science, we've heard where we're going, where we'd like to go, and I'm going to bring, I guess, an Australian perspective of how industry in this region, but also other areas around the world. So I do want to clarify, what I will talk about is not unique to Australia, but I guess how this science can be applied in your facilities in a practical setting to I guess, support not only whether you need to have a precautionary allergen labelling statement or not, but how this is a way your business can demonstrate due diligence. So I'm going to probably challenge your thinking a little bit as to sort of where this actually fits in the puzzle of the wider, I guess, risk mitigation processes that your businesses already have. And I will sort of finish off with touching a little bit on where analysis fits into all of this. So, you know, everyone in the room that is here representing a food business will be using analysis in some form or fashion to support all of their food safety processes that they have in place. And I think once we dive into the allergen space, we kind of sometimes lose a little bit of reasoning, or, you know, we know it's hard and complex, but hopefully I'll give you some things to consider there as well. So just a little bit about who the Allergen Bureau are. I know many of you in the room will have heard of who the Bureau are, but for many it may also be new. So the Allergen Bureau is a membership-based not-for-profit organisation. So some of you sitting in the room will see your company logos on this screen, I'm sure, and think, wow, I didn't know I was a member of the Allergen Bureau. But, you know, obviously regionally, your businesses, if your logo is there, does support the work that we do. We are completely funded by the food industry, and really were established in 2005, you know, 4 years after the Food Standards Code in Australia required mandatory allergen labelling. Industry got together and said, how are we actually all going to hold hands and work out how we're going to manage allergens and do some kind of quantitative risk assessment to support risk mitigation. So as Monique said, I'm representing the Allergen Bureau today. We have all of the directors provide our support to the Bureau in a voluntary capacity, and we couldn't do that if our businesses did not support us to do that as well. So a little bit about the VITAL program. So again, many of you may have heard about the VITAL program. You may hear that it's an online tool that's going to make life easy, and by all means it can and does. Again, this is a food industry, driven initiative. I really do want to kind of ground ourselves on where all of this started. So as I said, 2005, the Allergen Bureau sort of really came together. The very first concept of doing a quantitative risk assessment to support a precautionary allergen labelling statement actually started in 2007. Now, when we reflect, it wasn't probably the best science, I'm going to say, you know, but we had to start somewhere. And then, you know, some wonderful people before me that were involved in the Allergen Bureau in 2011 reached out to wonderful people like Renee Creval and our other VITAL scientific expert panel members to partner and determine how could we use clinical oral food challenges to try and navigate our way in a risk assessment process within the industry. And you can see, you know, we've had some different versions along the way. We're sitting at VITAL 4 now, with the current reference doses in the VITAL 4 version adopting the Codex-based reference doses. So, you know, there's a couple of points I want to sort of point out here as well, is that at least within the global space, it's the work of Renee and his colleagues that have really, I guess, opened the pathway for the wider FAO WHO expert panel consultation, and a lot of the outcomes that we see now sitting at Codex are because of the work that's been underpinned through the vital risk assessment process. In Australia, we're definitely seeing a change in the landscape. So I think back to Neil's point around regulatory adoption and enforcement, we've seen a shift change. Our enforcement agencies are now asking companies, how can you verify the fact that your PAL statement has actually been appropriately used on your products? And when consumers are reacting, you know, they're further interrogating us as businesses, I guess. So I just wanted, for those of you that are knee-deep in the weeds, right, of managing food safety, I do want to boil it down in a bit of a more simplistic approach. And I think, you know, whilst we talk about thresholds and the complexity around, you know, doing an allergen risk assessment, and having been there and done it myself, I completely understand. But I think we've got to start at the beginning. And the beginning really is, where are the allergens? How do we control them, right? Are you actually managing them appropriately? And I think, you know, whilst we can say we can do a quantitative risk assessment, really as businesses we need to be asking ourselves, what more can we do to actually further reduce the allergen risk in our business? So actually going through the process of performing a quantitative risk assessment allows you as your business to really critically challenge where your allergen risks are, and then allow you to then further try and reduce the risk. So that by the time you are doing a quantitative risk assessment, you should be managing at the lowest possible risk for your business anyway. We shouldn't then be having to use a PAL statement if it's not required, because you're already managing your risk profile. So think of that quantitative risk assessment as, you know, another tool in the toolbox, I guess, for how you can produce safe food, right, and protect the consumer. So, you know, thinking about the process that the Allergen Bureau has to I guess, take the science and distill it for industry, you know, we really kind of highlight, do that investigation, understand where your allergen risks are. Make sure that what you discover then feeds into your allergen management program. Make sure you get the right controls then to manage the risk that you identify. And only then can you actually do a quantitative risk assessment. And when you've done the quantitative risk assessment, you know, there's other questions you ask. I think to Neil's point, you don't stop. You then have to say, well, that doesn't sit well with my risk appetite. What more do I need to do? Because I don't like the outcome from my quantitative risk assessment. It's not I've done it, I put my PAL statement on the pack and off I go. And then obviously that last piece of the puzzle is, you know, the allergen communication. You know, the PAL is the last puzzle piece, right, for us as industry to be able to communicate to the consumer that there is a risk and they should avoid eating that food. And I will touch on my last slide, really from a consumer's perspective. They want to know which is the product without the PAL that's been risk assessed, which is a really different way of thinking for us in the industry. So again, thinking about everything that we've heard about the science, you know, and we think about how to distill that quantitative risk assessment, understanding where those cross-contact allergens are is obviously, you know, the first piece of the puzzle. And to Neil's point, you know, it does start at the beginning with our ingredient suppliers. So all of you that are ingredient suppliers, please do the quantitative risk assessment. It applies to you as well. You're just not going to tell your customer that they need to have a PAL statement, right? We need to know the number so that it can feed into the quantitative risk assessment that a packaged food operator is going to use. We've got our reference doses, right? They're now, you know, recommended and sitting, waiting at Codex to go through. From a reference amount perspective, that's how much does the consumer eat. And again, there's some great science and advice coming out there around, you know, it should be greater than the serving size, but, you know, should you be using a P50 or a P75 based on sort of population consumption data that's available. Once you've kind of plugged all of that in, you're going to know whether you have greater or whether you're above or below the reference dose, at which point you need to have a precautionary allergen labelling statement. I know it seems simple, and I know it's not simple, but again, you know, every step along the way is an opportunity for you as a business to ask how you further reduce risk, right? It's a risk mitigation tool. So, you know, our message to industry really is, it starts with an allergen management program. You can't leap in and do a quantitative risk assessment if you do not have a sound allergen management program in place. And then starting from there into your risk assessment, You're going to be able to then identify those allergens, focus on your prerequisites. I've done many HACCP risk assessments in my time, and when we look at in our HACCP tables and we come across an allergen and we go, how do we manage that in our facilities? We go, oh yeah, we do that through cleaning. Oh, that's a PRP. Move on. Next hazard. Every time you kind of dismiss to a prerequisite program as part of an allergen control, can you please go and interrogate it and find out that it's actually doing what you think it's doing? So our controls and where those risks are in different places to what we're used to, I guess where we're used to looking. Again, adopting the VITAL program. So everyone says, oh, you've got this fancy VITAL tool, it's a calculator. From an Allergen Bureau perspective, it's really about the program. Right, and that program, to Neil's point in his slides as well, is challenging yourself in that business to ask, can I eliminate, can I reduce my risk? And only once I've done that, how on earth am I going to manage that once I've accepted that risk profile? And then feed that into my quantitative risk assessment to work out if I need to communicate a risk to the consumer. And again, like my lovely colleagues have said, we really want industry to be in a position where we're only applying that precautionary allergen labelling statement when there's evidence, there's scientific evidence based on our risks in our facility to do so. Not because we've done a risk assessment and it's told us that we don't need a PAL, but we still feel like we really should. We're not helping the consumer when we go down that path. So where does analysis fit into all of this, right? And again, Renee touched on the fact that there's more harmonisation needed in this aspect as well. And I've sort of tried to unpack sort of the major areas where businesses would really sort of look to use allergen analysis to support what's happening. Ideally, obviously, it's there to help validate and verify your controls, not only from a quantitative risk assessment perspective, but also your allergen management programs. You can and you should, right, use analysis to support risk profiling of your ingredients. More importantly, I would say to check the suitability of the actual product or raw material to make sure that you can actually detect the allergen. There's lots of scenarios where based on the actual type of the food, matrix that it might be, or the way that it's been processed, that the protein can't actually be detected, right? So if you can't detect it, there's no point in using that as a marker in cleaning validation or verification. You'll get great negative results, but you won't know that you've actually got a risk. Obviously, we're using analysis for cleaning, making sure it's effective. There might be some kind of analytical testing that you're doing, say in a dry blending facility, where you might be purging, doing a product-to-product flush, for example. You might need to collect that flush solution to determine how much allergenic protein might remain. And that can then feed into a quantitative risk assessment. And again, you can use that data then to further challenge, well, should that number actually be lower? How much lower can I get it without impacting cost to the business? Our quantitative risk assessment inputs, and every time we think, we have an idea of what that risk is. We do need to sort of, I guess, dip our toe in the water and make sure that, we're close. So analysis is helpful to let us know we're in the ballpark. And I think that's the other thing we've got to remember that, and again, as Neil alluded from a risk assessment outcome perspective, the questions, allergen analysis it's not a precise, exact science. And we need to sort of go into analytical testing knowing that is the case. And if we get a number and it's in the ballpark of what we're expecting, then we're good. If it's 100 times higher, then we know we need to sort of go back and have a look. So you'll want to check your inputs so that your assumptions are going to be correct. You need to test your outputs of your risk assessment, and this is where industry is really uncomfortable. If you do a risk assessment and you find that you need to, whether you need a PAL statement or not, you find that you have your residual allergenic protein that remains in a finished product. Yes, it's uncomfortable, but you have to test it. Because if you don't test it, you won't know whether you're close to your expected theoretical risk assessment outcome. So I think it's really important that, as we look at this, analysis it has a part to play, but you can't grab a sample, test it, get a result, and say, I'm going to use that analytical value to determine whether I need to have a PAL statement or not. Because that would be the inappropriate use of analysis if we're going down that pathway. So just some considerations. we don't have standardised reporting. So I think that's really important for industry. And those of you obviously, in the space, managing, looking at reports for allergens or doing the testing yourself will know that, there's 3 different ways a milk protein report, can be issued. We need to be mindful that any kind of analysis that's being done is reported as total protein from the allergenic source in parts per million. And there's a lot of work now being done behind the scenes. So AOAC has developed a new guidance document for kit developers. So we now have guidance, really clear guidance for kit developers as to how results should be reported. So in the back end, we're now asking those guys that sell the kits to say, please, if you're making one, make sure that the results are reported like this, so that industry can just get the result and see how it compares to their quantitative risk assessments. We do need to be mindful that the questions that we need to ask ourselves as risk managers is the method suitable? So we know that anything that is going to impact our protein structure is going to impact the ability for us in the laboratory to be able to detect the protein. So I was saying to Neil before, normally my first question with my laboratory hat on will be to say, send me the sample that contains the allergen. And it's, no, I don't want your money. I just need to know that I'm going to be able to give you the right result. Because if I can't find the protein, you can't use that particular product for a cleaning validation. You need to find what else you've got. As I mentioned, it's not a precise science. We can't expect it to be. We're asking, that ELISA kit to do something that it unfortunately wasn't really designed to do. But again, we need to go into reading and looking at our results, knowing all of that. And again, analysis alone, as I said, is not suitable. If we think about the complexity of our manufacturing facilities, how cross-contact occurs, one sample from one batch is not going to give you, the appropriate outcome for your risk assessment. So again, when we're looking at where should we sample, we need to be thinking about, all the processing equipment that's involved to produce the product, not just the line. I often find people just test the sample off the line. So again, you've really critically got to be assessing where your risks are and using analysis, with its constraints in the appropriate way to support your risk assessment. There is a vital standard, so is there a solution for industry? The Allergen Bureau released a vital standard in 2019, so it is designed as an addendum to any GFSI audit. And this is really driven by the consumer. So the allergic consumer wants to be able to walk into a retail store and buy a product without a PAL statement and know that it's been risk assessed. So therefore it's a safer choice for them. So really that's what this standard is about. It's not designed to be make you jump through hoops. Really it's looking at the verification process that needs to be checked against your quantitative risk assessment, right? And then once you've got the standard, you may or may not use an on-pack mark. And again, that aligns very nicely with the recommendations that came out of the FAO WHO expert panel as well. So with that, I'm going to hand back to you, Monique.
Thank you very much. So I think, Jasmine, you made very clear how important it was for food business operators to set up a very robust and comprehensive allergen management program, integrating prevention, control, verification, communication. So I believe the 3 of you have made very clear how complex the topic was.
Yes.
And maybe it has raised questions in the audience. So I have some questions for you, but we would prefer to leave the floor to the audience and see if we have questions in it. Yes, we have someone here. Over there asking question 2.
A general question. Igor Berezhnoy, Food Safety Manager, Lactalis. Imagine we have a product where systematically we have a gluten level below 5 milligram. And it's proven it's like that in 100% of the cases. So what would be your recommendation? Should we make a PAL statement or not? Or should we limit it to just out-of-control situation for risk assessment? Because as we know, as the legislation is lagging, right now there is no limit, and we might be prosecuted if there is a case of anaphylactic reaction, even though all our assessment proves that it should not be the case.
Neil, maybe. Would you like to
Well,
take this one?
yes, there's a number of questions within that. So
There seems to be.
the topic of systematic presence of cross-contact allergen is hotly debated whether it's appropriate to use precautionary labeling in that circumstance. So we see, for example, in the recently proposed Dutch regulation, that the situations of systematic cross-contact would be excluded from precautionary labeling, and actually there would be an expectation that systematic cross-contact would appear within the list of ingredients. Other authoritative bodies or other stakeholders have got different opinions on how to manage systematic cross-contact. And we have had across the expert community discussions about having an alignment project on how to deal with that topic. So that there's a kind of outstanding issue around systematic cross-contact. In terms of what the level of cross-contact is, then quantitative risk assessment, if there is a good understanding of what that level is and how consistent it is, should enable you to make a good decision around what is the reality of risk experienced by the allergic consumer. And that should feed into your precautionary labeling decision. You mentioned there also about, I think you mentioned about, is there an upper level associated with when precautionary labeling is not appropriate? Or maybe I misheard, but that's also an area of hot topic as well. Are there situations where there is so much allergen that may be present that precautionary labeling is not a sufficient risk management measure to inform allergic consumers? So that's also a kind of unmet need in terms of alignment. But there are differences in regulations across the world in how to manage that sort of situation.
Yeah.
But
Very clear. Any adds from you, René?
No, I think Neil's understand perfectly well, actually. Thank you.
Okay.
Yes,
Quite complex question indeed.
it is, yes.
Yes, please, here. Second question. From Nestlé.
No microphone will come.
Okay,
Hi, John.
John Donaghy from Nestle. Thank you very much for the panel. It's a great discussion, and in particular, Neil has sort of outlined a lot of the, let's say, issues we have about alignment. My question is about how we get alignment with regulators in terms of that they will accept quantitative risk assessment. You know, when I give, maybe sometimes I position it as a sort of examples of situations that we've been in. You may remember the lecithin story from a couple of years ago with peanut, and there was a RASFF alert with very high levels of peanut. And in that case, we did a full quantitative risk assessment through from our supplier right through our manufacturing process. And we had used the material, and we did a quantitative risk assessment and didn't deem we did not need PAL. We had to confront that situation with many different authorities, and different authorities had different opinions as to our quantitative risk assessment. And we did that risk assessment using EDO1, so the most stringent means of quantitative risk assessment. And there was authorities, even two authorities within the same country, who had differing opinions. One wanted us to do a recall, and the others wanted us to, or were prepared to accept the quantitative risk assessment. So that is one element about using quantitative risk assessment, and the other element is around this alignment with regards EDO5 and EDO1. Because René has pointed out the majority of consumers. And we have in many situations, where we supply, products to more vulnerable, populations, whereby EDO5 may not necessarily be appropriate. Again, will that be acceptable across the different regulatory environments? As you say, we've seen now, for example, in the Dutch regulations, where they accept Codex, for example, and what will be the consequences if someone is selling within a country like that, but labeling at EDO1? So we are actually being stricter based on our own risk assessment. How would authorities deal with that situation? So I'm trying to give a couple of different examples of how this will play itself out, with regards to what we do in a regulatory, a challenging situation.
So highlighting a little bit more the complexity in the field. Do you want to build on this, René?
Well, I can say at least a few words because casting my mind back to my Unilever experience, yes, I very much sympathise because, yes, different authorities, I mean, Europe or the European Union is meant to have a single market, but from the point of view of allergen residues, it is definitely not a single market. I don't think it's now. So I think there's a lot of learning actually to be done that way actually as well. I think it's the only solution.
I probably can comment. Oh, sorry.
Please,
Hannah,
please,
am I allowed to?
Jasmine.
I can comment from an Australian regulatory and enforcement perspective. They can be trained. It takes a while, but we are now in a position where our enforcement agencies are looking and asking the question for products where consumers may have had a reaction to a product with or without a PAL. One of the first questions that is being asked is, please show us your quantitative risk assessment, and to support either whether the PAL statement should have been applied or not. The Allergen Bureau is hoping to publish a paper soon which will actually have a case study around a consumer that reacted to a product without a PAL. So, below the reference dose, they were in that percentage that's expected to have a reaction. They had a mild to moderate reaction as expected by the clinical data, and there was no action taken against the food manufacturer because they were able to demonstrate that the risk assessment had been performed. And and they had obviously data to support the fact that they were managing at that risk level.
Really.
So
So
there's
I
hope.
see-
There's hope.
Yeah, I see the time is over, unless there is a last burning question in the audience.
If we see any flames over there.
No, just the last one then. Yes, please.
Burning question. Oh, we need a microphone. I need a
Yeah, look, and I know we're sort of 2 minutes done. I think as an industry, we actively want to assess risk, manage risk, and communicate risk, right? It's kind of ingrained in us. That's what we're built to do as food safety managers. From a consumer perspective, consumers are confused. There are so many PAL statements, they don't know which one is the right one. They take risks, right? Which also does not help the food industry. But when you truly have a conversation with them and ask them around what the issues are around food availability and choice for them, they want to know which product has been risk assessed, so there's science applied, and that business can support the fact that a PAL statement is not required. So for them, that then speaks volumes around the company, the brand, the risk appetite, right? So, and honestly, that's why the standard was designed in conjunction with our patient advocacy groups in Australia. So it's really, as I said, it's the counter opposite to how we're built in the food industry as to meeting a consumer need.
Thank you, Jasmine. So please join me to thank Jasmine, Neil, and René for their insightful intervention.